Yes.
Yes.
Okay. Okay.
Yes.
Let's begin our today lecture. I now give you my presentation. Give my presentation. Sammy, please, can you listen presentation now?
Yes.
Yes. Okay. So, my dear colleagues, let's begin today's lecture. Today's lecture will be devoted again hormones, and today will be very important topic concerned to concrete hormone. Today we will talk about hormones of pancreas. Hormones of pancreas. My dear colleagues, you know histology, and you know what endocrine part of pancreas is very complex. It consists from different kind of cell, alpha, beta, delta, F cells, and other. And each cell of isle, of Langerhans isle of pancreas, specialized on production certain hormones. For example, alpha cell produce glucagon. Today we will talk about that hormone. Beta cell produce insulin. The most part of my lecture today will be devoted to insulin. Beta cell produce somatostatin. F cell produce pancreatic polypeptides, and so forth. So this endocrine gland produce a lot of hormone, a lot of biological active substances. But today, I want to begin my lecture since consideration, mechanism, effect, structure, and property of insulin. As I said you earlier, insulin produced by beta cells of pancreas. Insulin represents comparatively small protein which consists only from 51 amino acid residue, but in spite its low molecular mass, insulin is an oligomeric protein. In other words, it consists from two polypeptide chain, and that polypeptide chain bind to each other with covalent disulfide bond, as you can see on this scheme. Alpha chain shorter compared to beta chain. Alpha chain consists from 21, and beta chain consists from 30 amino acid residue. Now, I want to say you some words about synthesis of insulin. Synthesis of insulin in pancreas is very complex process. Initial precursor of insulin, uses special protein which called preproinsulin. This protein consists from only one polypeptide chain, and in their structure include 110 amino acid residue. And preproinsulin don't possess any biological activity. But that precursor undergo effect special proteolytic enzyme. This enzyme has a property like trypsin, resulted from preproinsulin lose 24 amino acid residue and transform into proinsulin. So, in other words, proinsulin product of restrict proteolysis of preproinsulin. It's enzyme catalyzed process. Proinsulin, like preproinsulin, it's a protein which consists from only one polypeptide chain. This polypeptide don't possess any property of hormone. It's not biological active peptide. But proinsulin later may undergo transformation into insulin. And again, synthesis of active insulin from proinsulin coupled with restrict proteolysis. Special trypsin-like protease undergo proteolytic attack polypeptide chain of proinsulin in these two places, one place and two places, resulted from molecule of proinsulin undergo breakdown to yield end product. That product will be denominated as so-called C-peptide, and other part of molecule, which, as you can see, consists from two polypeptide chain binded with disulfide bond, represented as insulin, active insulin. C-peptide is not biological active substances. It don't possess any property of hormones. It's side product by synthesis of insulin. And insulin, excuse me, I accept all other students. So let's carry on. And insulin is active hormone. Insulin is active hormone. So, my dear colleagues, now about property of insulin. After synthesis in beta cell, insulin undergoes storage in special so-called secretory granule of insulin. Secretory granule of insulin contain represented preferentially as hexamer, each contain in their structure 3 molecules of active insulin and 3 molecules of C-peptide. In other words, side product biosynthesis of insulin includes structure of secretory granule. And my dear colleagues, please note, secretory granule components stabilized here in granule form special specific crystal with zincum cation. So in other words, secretory granule of insulin granule consists from insulin, the same or equimolar amount of C-peptide and zincum. It's storage form of insulin. Now, let's say about regulation, secretion, and synthesis of insulin. My dear colleagues, then you talk about regulation, secretion, and synthesis of insulin. Please pay attention. What most important control factor for secretion and production of insulin is level of glucose in the blood. Increase glucose in the blood appear hyperglycemia, bring to increased production of insulin by synthesis of insulin, and simultaneously increase secretion of insulin. So about production, my dear colleagues, beta cell membrane, plasmatic membrane, plasmalemma, contain special glucose transporter, GLUT2, and then concentration glucose in the blood increase, arise hyperglycemia. Glucose penetrate inside beta cell, and here glucose in cytoplasm of beta cell uses as activator special factor transcription, which take part in the activation expression gene of insulin. In other words, then glucose penetrate in cytoplasm of beta cell, it's bring to activation expression insulin gene, resulted from increased synthesis of insulin. From other side, then then glucose penetrate in cytoplasm of beta cell. Here, glucose bring to increase process its catabolism in the glycolysis. The major pathway glucose catabolism in beta cell, it's breakdown in glycolysis. Glycolysis, I hope you know, it's process which provide cell with energy, such as during glycolysis exist two reaction substrate level phosphorylation, which bring to synthesis ATP. In other words, increased concentration glucose in beta cell bring to increased concentration ATP inside cytoplasm of that cell, resulted from appear depolarization of membrane of beta cell. Again, it's bring to activate open so-called voltage dependent ion channel in membrane, resulted from calcium outside cell penetrate in cytoplasm, resulted from firstly accumulation glucose in cytoplasm, in second bring to increase concentration calcium in cytoplasm of beta cell, and then calcium concentration in cytoplasm increase, it's bring to stimulation process secretion of insulin granule into the bloodstream. So, my dear colleague, in other words, then arise hyperglycemia. For example, it may be after eating, after fooding, resulted from arise hyperglycemia, and hyperglycemia bring to increase production of insulin and its secretion in the blood. My dear colleagues, please note, in regulation secretion of insulin is very important too. Epinephrine interact with so-called alpha-2 adrenal receptor on membrane of beta cell, resulted from decreased secretion of insulin. In other words, stimulation of sympathetic nerve system, stimulation of production of epinephrine bring to decreased process secretion of insulin. From other side, stimulation of parasympathetic nerve system bring to activation, increasing secretion of insulin. So it's about regulation, secretion, and production of insulin. Now, about role of insulin in the blood, fate of insulin in the blood. My dear colleagues, please note, insulin secreted in the bloodstream simultaneously with C-peptide. But C-peptide don't possess any biological activity. But if you take into account what secretion of insulin in C-peptide and insulin in C-peptide in equimolar amount, it may be uses determination concentration of C-peptide for evaluation secretory function of beta cell. It often take place in a diagnosis different diseases. So insulin time life in the blood about 10 minutes. In the 10 minutes, insulin may interact with its receptor. Now we will say about receptor. Or insulin may undergo process catabolism. or clearance. Clearance of insulin, its decomposition, take place in the liver and in the kidney. So liver cell, hepatocyte, and kidney cell undergo absorption insulin from the bloodstream, and here it's undergo effect proteolytic attack by enzyme insulinase. Insulinase is a syncytin-containing proteolytic enzyme which catalyze process breakdown insulin to yield free amino acid. And please note, insulinase take part not only in clearance of insulin. This enzyme may take part in the catabolism of glucagon 2 and other peptide hormones. So now let's return to target of insulin. Insulin is hormone. For that reason, it must interact with may penetrate in target organ and affect bind here with a specific insulin receptor. First of all, about target organs for insulin. My dear colleagues, about all visceral organs may respond to effect of insulin. For that reason, a receptor for insulin localizes absolutely in all target cell of target organ in the human body. Now about receptor. Receptor for insulin has a complex structure. You must know what active insulin receptor represented as tetramer. It consists from 4 polypeptide chain, and this 4 polypeptide chain may be subdivided into 2 different kind of polypeptide chain, 2 alpha and 2 beta polypeptide chain. Beta polypeptide chain insert in the membrane, plasmalemma of target cell. It's membrane binded peptide protein. Alpha subunit or alpha chain, which create alpha subunit of insulin, don't bind with cell membrane, but they bind with beta chain. Alpha and beta chain bind to each other with usually disulfide bond. And different kind of chain possess different property. Alpha subunits, alpha chain of insulin receptor contain specific site binding of insulin. Beta chain possess catalytic property. They possess property activity of tyrosine kinase. So, in other words, beta chain of insulin receptor may catalyze process phosphorylation of protein. My dear colleagues, previous lecture we said about protein kinase. Protein kinase A, I said you about. Protein kinase C, protein kinase G. That kind of protein kinase catalyze process phosphorylation protein, but for phosphorylation uses residue amino acids such as serine and threonine. Beta subunit of insulin receptor may catalyze phosphorylation of protein only binding residual phosphoric acid with tyrosine residue, which includes structure of polypeptide chain. For that reason, that subunit of receptor possess so-called tyrosine kinase activity. My dear colleagues, please note, insulin receptor may consist in two different kinds. First, in rest condition, then in the blood absent the molecule of insulin, insulin receptor undergo dissociation resulted from tetramer, undergo dissociation to yield two dimer. Each dimer consists from alpha and beta subunits. Beta subunits in that condition don't possess any catalytic property. But then insulin appear in the blood. It's bring to binding insulin with one alpha subunits resulted from take place association to dimer of insulin receptor in tetramer, and receptor become active. So then receptor represented as tetramer and bind with hormone, create hormone receptor complex. Beta chain of insulin, beta subunits of insulin get catalytic property and begin catalyze tyrosine kinase reaction, undergo certain protein inside the cell phosphorylation. So it's about structure and property of insulin receptor. Now, my dear colleagues, let's say about effects of insulin on metabolic processes. All effects of insulin may be subdivided into two separate groups. Very fast effects which appear immediately after binding insulin with receptor. Very fast effect of insulin may be coupled with increased permeability rate for glucose. Second kind effect of insulin, fast effect, appear in some minutes hormone receptor complex, and fast effects coupled with change activity of enzymes, and that change activity of enzymes provided by formation of hormone insulin receptor complex. Next kind of effect, slow effect of insulin appear in usually in some hours after formation insulin receptor complex, and that kind of effect coupled with induction or repression enzymes in the target cell. And exists also one effect, typical effect of insulin, very slow effects. It's so-called growth effects. These effects appear usually after a day or some day even, and that effect coupled with change of control of growth of cell differentiation, apoptosis, and so forth. My dear colleagues, let's say about mechanism appear all that effect of insulin. Mechanism it's appear. Please see, my dear colleagues, appear effect, different kind of effect of insulin coupled with existence two major signal pathway inside the cell coupled with insulin. And my dear colleagues, first pathway, please note, first pathway coupled with activation so-called RAS protein, which bring to activation mutagen activated protein kinase signaling pathway, and that pathway coupled with regulation process expression of genes, regulation production of protein and nucleic acid, and this signal transduction pathway coupled with insulin take part in the control cell growth, proliferation, apoptosis, and so forth. Second signal transduction pathway coupled with insulin, coupled with activation special enzyme, phosphatidylinositol triphosphate kinase. And this metabolic pathway, signal transduction pathway, coupled with insulin, coupled with providing different kind of fast and very fast effect of that hormones. Now let's say concretely about each signal transduction pathway of insulin in target cell. First of all, let's say about effect of insulin coupled with activation of RAS protein. My dear colleagues, what you have to know about that signal transduction pathway. Then insulin bind with insulin binding site on alpha subunits and create active hormone receptor complex, beta subunits of receptor for insulin, get tyrosine kinase activity. First of all, beta subunits undergo phosphorylation each other. So that subunit undergo phosphorylation other subunit of receptor, residue of tyrosine in that subunit undergo phosphorylation. From other side, that active beta subunit bring to phosphorylation that beta subunits, other subunits of insulin receptor. In other words, first effect of insulin coupled with phosphorylation beta subunit itself. Then beta subunits undergo phosphorylation, they get ability bind special kind of cytoplasmic protein. And that cytoplasmic protein uses as adapter protein of that hormone. Protein HSHC immediately bind with phosphorylated beta subunits of insulin receptor. After that association, SHC bind with other proteins such as GRB-SOS. And after formation of that complex get ability to bind specific GTP binding protein RAS. RAS bind GTP and in the complex with other that kind of protein binding with beta subunits get catalytic property, and resulted from its bring to activation special so-called RAF protein. RAF protein under the activation too resulted from this kinase, this enzyme undergo phosphorylation other protein, but phosphorylation other protein not via phosphorylation tyrosine. It's take place via phosphorylation threonine or serine only. Resulted from undergo activation cascade of mutagen activated protein kinase. And mutagen activated protein kinase bring to phosphorylation of specific factor of transcription, resulted from inside cell increase, first of all, gene expression, certain protein, and synthesis mRNA that latter uses for process translation resulted from inside cell synthesis specific spectrum of protein. And my dear colleagues, please note, first of all, this protein may take part in the control of growth of cell, differentiation of cell, control apoptosis, and so forth. So they take part in the formation, so-called very slow effects of insulin, growth effects. But from other side, in this signal transduction pathway coupled with insulin may increases process expression gene certain enzymes resulted from increased production of that kind of enzymes. For example, insulin bring to induction synthesis of glucokinase, pyruvate kinase, ATP citrate lyase, FAS, fatty acid synthase complex, and so forth, and so forth. For that reason, insulin bring to appear slow effect for glycolysis, oxidative decarboxylation of pyruvate, synthesis of lipids, pentose phosphate pathway. It stimulates all that processes, as you can see from that table. And exists special enzyme. It's enzyme called phosphoenol pyruvate carboxykinase. Gene of that enzymes undergoes suppression by insulin. For that reason, simultaneously, insulin brings to inhibition process gluconeogenesis in the liver. And all that effect coupled with change of production enzymes, which take part in carbohydrate and lipid metabolism, appear in some hour after effect of insulin. So it's about one effect of insulin what provided for activation of RAS protein in cytoplasm of cell. Now about second signal transduction pathway, which take part in the regulation metabolic effect of insulin inside cell. This signal transduction pathway coupled with effect on special enzyme which designate as phosphatidylinositol triphosphate kinase. Let's say about that signal transduction pathway coupled with insulin. My dear colleagues, then we talk about activation of RAS protein. We said what active insulin receptor undergo phosphorylation, special protein which called SHC, which uses as substrate of receptor. But maybe other uses other substrate of insulin receptor. In metabolic pathway, in signal transduction pathway coupled with activation of that enzyme, as substrate of insulin receptor uses special protein which called insulin receptor substrate 1, IRS1. After phosphorylation of beta subunits of insulin receptor get ability bind of IRS and undergo its phosphorylation. After its phosphorylation, IRS immediately bind other kind of protein, GRB2, NCK, SYP, and the other. And after formation of that complex binded with beta subunit of insulin receptor, that complex get ability bind enzyme phosphatidylinositol triphosphate kinase. And binding of that enzyme bring to its activation. Phosphatidylinositol triphosphate kinase consists from two subunits. It's allosteric subunits, regulatory subunits, smaller, and catalytic subunits. After binding with complex proteins with IRS, after phosphorylation of IRS, enzyme get activity, get catalytic property, resulted from its begin catalyze reaction phosphorylation phosphatidylinositol 4,5 bisphosphate. Phosphatidylinositol 4,5-biphosphate. My dear colleagues, please note, these substances, it's phospholipids, which include structure phospholipid bilayer of a cell membrane. Membrane what includes structure insulin receptor 2. After phosphorylation by in reaction catalyzed that enzyme phosphatidylinositol 4,5-diphosphate, it's transformed into phosphatidylinositol 3,4,5-bis-3-phosphate. Appear residual phosphoric acid also and third carbon in molecule residue of inositol in molecule of that phospholipids. Resulted from inside membrane appear after activation of that enzyme, appear phosphatidylinositol 3,4,5 3-phosphate. And my dear colleagues, it's very important to know what that substances uses for binding special cytoplasmic enzyme, which called phosphatidylinositol 3-phosphate dependent kinase, PDK. After formation that phospholipid in lipid bilayer, that enzyme, that kinase, bind with membrane, and binding of that enzyme with membrane bring to its activation. And it's very important to know what phosphatidylinositol 3-phosphate dependent kinase catalyze reaction phosphorylation of different protein in cytoplasm too. And as one target of that enzyme, one substrate of that enzyme, uses protein kinase B. We know protein kinase A, protein kinase C, protein kinase G, tyrosine kinase. So, my dear colleagues, let's get started. Let's continue our conversation. I return to my slide. So, in other word, excuse me. In other word, this signal transduction pathway coupled with insulin bring to activation protein kinase B, and all fast effect of insulin, fast effects and very fast effect, immediately effect of insulin, coupled with activation that enzyme. Why is it? My dear colleagues, this enzyme, that protein kinase, maybe uses different substrate, A substrate of that enzyme maybe uses such enzymes as protein phosphatase, maybe uses protein phosphatase, catalyze cyclic AMP. It's so-called phosphodiesterase cyclic AMP, phosphodiesterase cyclic AMP. And as substrate of that enzyme maybe uses a special kind of glucose transporters. My dear colleagues, please note, as substrate of protein kinase B maybe uses glucose transporter 4, and glucose transporter 4, it's insulin-dependent glucose transporter. For that reason, then increase secretion of insulin, this kind of glucose transporter undergo phosphorylation, and it's bring to increase permeability membrane of that insulin-dependent tissue, adipose tissue and skeletal muscle tissue and myocardial tissue for glucose. First of all, I want to remind you information about glucose transporter. Exists different kind of glucose transporter. Insulin affect only for one glucose transporter, GLUT4, so-called, which localized, as I already remind you, in adipose tissue and skeletal muscle cell, cross striatic muscle cell. All glucose transporter represented as a protein which consists from lot of so-called alpha helix, which provide insertion of glucose transporter in cell membrane, and cross that glucose transporter may pass glucose toward its low concentration. So then concentration glucose in the blood increase, they will be moved inside cell. And please note, GLUT4 may exist inside cell in two conditions. First condition, it's dissolved in cytoplasm. Such glucose transporter don't take part in the transport of glucose. But that glucose transporter may insert in the cell membrane and take part in the transport glucose cross cell membrane. Please note, flow glucose cross cell membrane will be higher than concentration of glucose transporter in cell membrane will be more. So insertion, additional amount of GLUT4 in cell membrane bring to increase permeability cell membrane for glucose. And please note, in order to GLUT4 get ability insert cell membrane, it must be undergo phosphorylation. And phosphorylation of glucose transporter coupled with reaction catalyzed by protein kinase B. Protein kinase B, active protein kinase B, bring to phosphorylation dissolved in the cytoplasm of target cell, muscle cell or adipocytes, myocytes or adipocytes. GLUT4 resulted from its transform and insert in cell membrane resulted from sharply increased permeability cell membrane for glucose. And that effect appear in some second after formation of insulin receptor complex. It's a very fast effect, which appear about immediately after secretion of epinephrine, insulin. So other substrate of protein kinase B, as I said you already, maybe uses phosphodiesterase of cyclic AMP and protein phosphatase. Both that enzymes undergo active protein kinase B phosphorylation resulted from its undergo activation. Activation of phosphodiesterase bring to increase transformation cyclic AMP into uncyclic AMP in AMP inactive. And activation of protein phosphatase bring to dephosphorylation of enzyme, resulted from change activity of enzyme which undergo control this process phosphorylation, dephosphorylation, resulted from a rise cell response to effect of insulin. My dear colleagues, please note, glycogen synthase, pyruvate kinase, phosphofructokinase, pyruvate dehydrogenase, and acetyl coenzyme A carboxylase, that kind of enzyme active in dephosphorylated form. And insulin, please note, bring to activation protein kinase B, and activation protein phosphatase resulted from all that enzymes undergo dephosphorylation, resulted from they get higher activity, resulted from insulin, increase glycogen synthesis, increase glycolysis, increase oxidative decarboxylation of pyruvate, increase lipogenesis. But dephosphorylation in that pathway regulation, dephosphorylation, such enzyme as hormone sensitive lipase or fructose 1,6-bisphosphatase or phosphorylase A will be bring to inhibition process lipolysis, inhibition process gluconeogenesis, and inhibition process glycogenolysis. So all that effect from insulin appear in some minutes after secretion of insulin, and appear all that fast effects coupled with effect insulin for metabolic processes via activation of phosphatidylinositol triphosphate kinase signal transduction pathway. So, my dear colleagues, it's about metabolic effect of insulin. Here you again may see some information about effect of insulin for certain processes, permeability of cell membrane for glucose, induction of glucokinase, and so forth. We already said about that process. And my dear colleagues, please note that effect, that kind of effect, fast effect, is in the basis of reciprocal regulation glycogen metabolism. Glycogen metabolism in situations and increase secretion of insulin bring to activation of glycogen synthesis, increased rate of glycogen synthesis via activation of glycogen synthase D, glycogen synthase A. And insulin simultaneously bring to transformation phosphorylase A in phosphorylase B, resulted from increasing biosynthesis of glycogen, simultaneously accompanied by decreased process breakdown of glycogenolysis. So it's about metabolic effect of insulin for carbohydrate metabolism. Again, I want to summarize about effect of insulin for carbohydrate metabolism. My dear colleagues, then you said, answer that question about effect of insulin for carbohydrate metabolism. First of all, you must say about hypoglycemic effect of insulin. So secretion of insulin bring to decrease concentration decrease concentration glucose in the blood, decrease concentration glucose in the blood. And you must explain why insulin provides hypoglycemic effect. In the basis of hypoglycemic effect, there is increased permeability of cell membrane for glucose. Glucose penetrate inside target cell, and here uses for synthesis of glycogen, increase process biosynthesis of glycogen by insulin. Insulin increase process catabolism of glucose in glycolysis. It's bring to decrease concentration of glucose too. And insulin increase process pentose phosphate pathway utilization of glucose. So, in other words, insulin from one side bring to transport glucose from the blood inside cell, and inside cell it's increase rate processes glycolysis, synthesis of glycogen, pentose phosphate pathway, which coupled with utilization of glucose. From other side, insulin bring to inhibition process biosynthesis of endogenous glucose during process gluconeogenesis. All that effect of insulin is in the basis appear hypoglycemia. But you must know about effect of insulin not only for carbohydrate metabolism. You must know about effect of insulin on lipid metabolism too. What you have to know about that aspect effect of insulin? First of all, activation of biosynthesis fatty acid, activation biosynthesis of cholesterol, phospholipids is in the basis of increasing lipogenesis by insulin. From other side, insulin bring to inhibition process lipolysis inside tissue. Inhibition of lipolysis in tissue coupled with dephosphorylation of hormone-sensitive lipase inside tissue. About effect of, and also very important about lipogenesis, effect insulin on lipogenesis. It may be indirect effect coupled with providing adipocyte with glucose, such as increased permeability membrane of adipocyte for glucose. Glucose penetrate in adipose tissue cell, and here undergo transformation into lipids. It's other effect of insulin which bring to increased process lipogenesis in adipose tissue. Moreover, insulin increase biosynthesis of protein. We said will take part in the increase biosynthesis, different protein, different enzymes inside cell. And that effect of insulin coupled with its effect for mitogen activated protein kinase cascade inside cell, and activation factor of transcription. And simultaneously with induction synthesis of protein, insulin increase biosynthesis of nucleic acid. Preferentially, we said about RNA. So it's about effect of insulin for different metabolic processes in target cell. Or it's increased absorption of not only glucose, it's increased absorption of amino acid, and it's very important in the increasing biosynthesis protein provided by insulin in target cell. Moreover, its take part in activation sodium-potassium ATPase resulted from its bring to depolarization certain tissue, for hyperpolarization certain tissue, for example, myocardium, and so forth. So, in other words, effects of insulin is very plentiful. So let's stop say about insulin and let's begin discussion about other hormone what synthesizes in the pancreas gland. It's glucagon. As I said you, glucagon produced by alpha cells of Langerhans cells of pancreas. Alpha cell produce precursor of glucagon, pre-proglucagon. Its activation, transformation of prohormone into active hormone is the same as insulin 2. Pre-proglucagon undergo restrict proteolysis, transform into proglucagon. Proglucagon again undergoes restrict proteolysis, undergo transformation into proinsulin, proglucagon, and proglucagon again undergo process restrict proteolysis to yield active glucagon. Active glucagon represented as peptide which consists from 29 amino acid residues. So, what can you say about stimulation secretion of glucagon? Stimulation, it's very important again in stimulation secretion of glucagon, concentration level glucose in the blood. Decrease level glucose in the blood, decrease hypoglycemia, bring to increase production of that hormone, increase production of that hormone. In other words, that hormone designated as hormone of fasting. Its production increased during fasting or starvation. Then concentration glucose in the blood sharply decreased. After secretion, glucagon pass into the bloodstream and move to its target organ. The most important target organ for that hormone, it's liver and adipose tissue. So the major target organs for glucagon, liver and adipose tissue. What can you say about mechanism? Its mechanism, the mechanism regulatory effect, coupled with activation of adenylate cyclase inside cell and synthesis cyclic CAMP as a second messenger of that hormone. So, my dear colleagues, I don't describe all that mechanisms such as we said about adenylate cyclase mechanism in the effect of different kind of hormones. Now, I want only say then hormone glucagon bind with glucagon receptor inside target cell, undergo activation enzyme adenylate cyclase, and adenylate cyclase catalyze reaction hydrolysis ATP to yield cyclic AMP. And cyclic AMP uses as a second messenger of that hormone. Hormones from that hormone don't penetrate inside cell, but formation of a hormone receptor complex bring to accumulation cyclic AMP. And cyclic AMP uses as allosteric activator enzyme protein kinase A, which bring to phosphorylation different kind of protein, and phosphorylation different kind of that protein different kind of that protein bring to appear cell response. So it's cyclic AMP structure. We said about that. My dear colleagues, in other words, protein kinase A after activation undergo different its substrate phosphorylation. Phosphorylation, different protein in cytoplasm of cell coupled with binding residue of orthophosphoric acid to residue of serine and threonine in polypeptide chain, resulted from polypeptide chain appropriate protein become phosphorylated. Different kind of enzymes exist which use phosphorylation, dephosphorylation for control for their catalytic property. For example, phosphorylation of glycogen synthase, pyruvate kinase, phosphofructokinase, pyruvate dehydrogenase, acetyl coenzyme A carboxylase bring to inhibition of that enzyme. For that reason, glucagon bring to decrease synthesis of glycogen, decrease rate of glycolysis, decrease rate of oxidative decarboxylation of pyruvate, decrease rate of lipogenesis. From other side, phosphorylation of hormone-sensitive lipase, fructose 1,6-bisphosphatase, and phosphorylase A bring to its activation. For that reason, glucagon bring to activation increase rate of lipolysis in tissue, increase rate of gluconeogenesis, preferentially in liver, and increase rate process glycogenolysis in the liver. Resulted from result of all that effect will be increased concentration glucose in the blood, increase secretion glucose into the blood. As a result, activation process glycogenolysis, activation process of gluconeogenesis. And it's very important to know about regulation process by synthesis glucose during gluconeogenesis by that hormone. Why glucagon bring to activation of gluconeogenesis? From one side, it's bring to activation key enzyme of gluconeogenesis, increase activity for fructose 1,6-bisphosphatase, increase activity of pyruvate carboxylase and other enzymes. Simultaneously, that process gluconeogenesis require a high amount of energy for synthesis endogenous glucose, and as for production of energy for that process uses increasing process lipolysis. Lipids undergo process breakdown in fatty acids involved in the process beta oxidation release a lot of energy which undergo accumulation in ATP, and ATP involved in the process gluconeogenesis. From other side, glucagon bring to activation process proteolysis, resulted from release amino acid, and that amino acid may involve in gluconeogenesis too. So, result effect of glucagon, it's hyperglycemic effect. And hyperglycemic effect of that hormone coupled with activation process glycogenolysis and stimulation process gluconeogenesis. gluconeogenesis. So it's about effect of glucagon, effect of glucagon. Now, my dear colleagues, today I again must return to diabetes mellitus. I already said you some word about that disease in previous semester. Then we will talk. We have talked about the disorder carbohydrate metabolism. Now I want to remind you some aspects of diabetes mellitus. Diabetes mellitus may be subdivided in two major types. Type 1, insulin dependent, and type 2, insulin independent. Type 2, insulin independent diabetes mellitus, coupled with appear insulin resistance. Is insulin resistance. What's mean insulin resistance? Insulin resistance manifestation coupled with decreased affinity of insulin receptor to hormone insulin resulted from even normal secretion of insulin cannot bring to activation appropriate signal transduction pathway coupled with insulin and appear clinical manifestation of diabetes mellitus. Previous semester we said about causes of diabetes mellitus type 1 and type 2. We said about contingent of patient, age contingent of patient. For that reason, I don't return to that question. I want to remind you clinical manifestation of diabetes mellitus, preferentially type 1. The most prominent symptoms, hallmark symptoms, it's hyperglycemia, it's maybe glucosuria, it's uria, it's toasty, and decrease of body mass if we talk about diabetes mellitus type 1. Diabetes mellitus type 2 coupled with increased body mass, such as obesity is one very important cause appear diabetes mellitus type 2. So it's about clinical manifestation. Now about change metabolism at diabetes mellitus. Let's say about diabetes mellitus type 1. The most, the hallmark symptoms of diabetes mellitus type 1 is hyperglycemia. Simultaneously increase rate of glycogenolysis, decrease rate of biosynthesis of glycogen, decrease rate of glycolysis, increase rate of gluconeogenesis preferentially in the liver, increase rate of lipolysis, increase rate biosynthesis of ketone bodies, aceton for example, increase rate of proteolysis, and appear disorder in the water and electrolyte metabolism, appear water and electrolyte misbalance in the human body. Resulted appear that effect may be appear different complication of diabetes mellitus. We said about complication of diabetes mellitus previous semester, but I want to remind you acute complication of diabetes mellitus manifested by appear diabetic coma, unconscious condition which maybe bring to death of patient if we on time don't help patient with medicine. So exists different kind of diabetic coma, hyperglycemic, hyperosmotic coma. This kind of coma coupled with accumulation glucose in the blood resulted from increased osmotic pressure of the blood. Lactacidic coma coupled with increasing process anaerobic glycolysis in the body such patient. And ketoacidic coma is which coupled with increased biosynthesis of ketone body in the liver such patient. Please note, ketoacidic coma may appear as complication only at patient with diabetes mellitus type 1. It was conversation about acute complication of diabetes mellitus, but you must know about existing so-called late complication of diabetes mellitus. As late complication of diabetes mellitus may appear injury of different organ and tissue, as resulted from arise angiopathy, microangiopathy, microangiopathy. It may be nephropathy coupled with injury of kidney. It may be neuropathy coupled with injury function of brain, usually coupled with injury of myelin, function of myelin nerves. It may be retinopathy. It may be cataract. It may be cataract. And my dear colleagues, please recall what appear all that late complication coupled with increasing accumulation glucose in the blood and in the tissue. So increasing concentration glucose appear hyperglycemia, bring to appear specific non-catalytic pathway utilization of glucose. This pathway coupled with interaction with glucose with protein preferentially. Molecule of protein, polypeptide chain of protein contain in their structure different amino acids. And some amino acids contain in their structure nucleophilic functional group, for example, amino group or sulfhydryl group. And this functional group may interact with glucose, may take part in the binding of glucose. And for that binding, don't need any catalyst. It's in catalytic process. See here, then concentration of glucose inside cell or in the blood increase, glucose may bind with protein resulted from a rise so-called glycosylated proteins. Residue of glucose bind with polypeptide chain via Schiff-base bond. Schiff-base bond. It's tight bond. It's tight bond. And please note, my dear colleagues, formation of glycosylated protein bring to change conformation of polypeptide chain, its polypeptide chain. And conformation of polypeptide chain from other side bring to change property of that kind of proteins. Resulted from in the blood vessel, in the kidney, in the retina of eye, in the myelin, appear glycosylated protein, and this glycosylated protein cannot perform their function normally, resulted from a rise injury function of that organ. Arise if that kind of proteins appear in the connective tissue between cell in the endothelium of blood vessel, arise angiopathy. In the basal membrane of kidney, arise nephropathy, and so forth. Moreover, glucose may bind with hemoglobin, the one of most commonly protein in the human body, localizes in the blood, in red blood cell. It's hemoglobin. Then concentration of glucose in the blood sharply increase. In the same manner, undergo glycosylation resulted from a rise specific rate of hemoglobin, so-called glycosylated hemoglobin or glycated hemoglobin, other name, hemoglobin A1C, its acronym. And this kind of hemoglobin change property, so it cannot effectively take part in the transport of oxygen in red blood cell. From other side, accumulation that kind of hemoglobin may be used for diagnosis diabetes mellitus. And modern period of time now, quantification of glycosylated hemoglobin uses as one of most specific biochemical tests in diagnosis of diabetes mellitus. Next, glucose, then its concentration inside cell sharply increased, may be involved in unusual processes utilization. For example, then concentration glucose in cytoplasm at patient with diabetes mellitus. Enzyme aldose reductase transformed to reduction carbonyl glucose, resulted from glucose transformed into alcohol sorbitol. Sorbitol is osmotic active substances, and its accumulation bring to increase osmotic pressure in cytoplasm of cell. And the accumulation of sorbitol is very important in the injury function in the lens of eyes, resulted from a rise cataract, such as appear turbidity of lens. So it's also one unusual way utilization of glucose